This film demonstrates the essential steps in Briggs and King's procedure for nuclear transplantation in the leopard frog. The aim of nuclear transplantation experiments is to see whether the nucleus from one of the cells of a developing frog can function in place of the original zygote nucleus in the fertilized egg. First, the sequence of operations is explained diagrammatically, and then the practical details are discussed. On entering the egg, the sperm makes a small injury or prick. This induces the egg to start developing. This activation may be simulated by pricking with a fine tungsten needle. Artificial activation is the first step in preparing an egg for nuclear transplantation, triggering off development without inducing an unwanted sperm nucleus. Following activation, the egg nucleus migrates to the surface of the egg, where it may be found for a brief period as a small black spot. The second step in the preparation of the host egg is a surgical removal of the black spot, enucleation, shown here in section. The third step in nuclear transfer is the preparation of the donor cells. An isolated fragment of tissue, usually taken from an embryo, is placed in a disaggregating fluid, which causes the cells to fall apart so that they can be handled individually. Finally, a single cell is taken up in a pipette and injected into the previously activated and enucleated egg. A female frog is induced to ovulate by injections of frog pituitary glands. The mature, unfertilized eggs collect in the oviducts and are obtained by gentle squeezing. These unfertilized eggs already have produced the first polar body, but require the stimulus of activation before they can complete the second meiotic division. It is convenient to place the eggs individually on a slide with the pigmented or animal half up. The egg is protected by two coats. It is under slight pressure inside the vitelline membrane, which is the thin, tough inner coat, and is surrounded by an outer, thicker layer of jelly. A fine tungsten needle is sterilized by flaming, and the egg is activated by pricking. To prevent the eggs from drying out whilst waiting for the black spot to develop, they are covered by a physiological saline. The pink color is due to phenol red, which is a pH indicator. The activated eggs are now left for about 20 minutes to allow the second meiotic division to proceed. Activation having stimulated the second meiotic division to take place, the nucleus will now lie just beneath the surface somewhere near the animal pole. It can be located for a few brief minutes as a minute black spot, and intense oblique illumination is required to pick it out. Seen here in the center of the screen, the black spot is less than 1/1000th of an inch across. It is really a window into the interior of the egg. The transparent nucleus in metaphase having risen to the surface, interrupting the superficial layer of pigment granules. To enucleate the egg, the fine tungsten needle is inserted obliquely beneath the black spot, drawn upwards through it and out of the egg. The vitelline membrane is ruptured, and as the egg is under internal pressure, the nucleus and some of the cytoplasm are pushed out. As soon as the tension is released, the vitelline membrane springs back, preventing any further outflow, and the nucleus is effectively isolated. The enucleated host eggs are now left for a further half an hour, during which time the donor cells are prepared. These day-old embryo are early gastrulae. The jelly coat around the embryos is taken off, and the vitelline membrane, still protecting the egg, is carefully removed. The embryo, no longer protected by its membranes, may now be dissected with tungsten needles which have been sterilized by flaming. The embryo at this stage contains about 30,000 cells arranged in the form of a hollow ball. Flaps of tissue from the pigmented or animal half of the embryo are folded back to uncover the endoderm cells, which will later form the gut. The endoderm cells are the large pale cells which form most of the non-pigmented half of the embryo at this stage. A little piece of endoderm is cut out for disaggregation. A pipette is sterilized by sucking up boiling water and cooled in sterile saline. A few drops of disaggregating medium are put into a small chamber and the fragment of endoderm transferred into it. The disaggregating medium lacks calcium and magnesium and causes the cells to separate after about 15 minutes treatment. The micropipettes for injecting the nuclei may be made in a Livingston pipette puller. A piece of glass tubing, 1 mm in diameter, is held under tension between electrically heated elements. Current is switched on, the glass softens, and the spring tension is released. The pipettes are taken from the machine with their ends sealed, and the fine shaft must be broken under a microscope until a well-shaped tip is achieved. The aim is to achieve a somewhat oblique break, with the end sharply pointed to allow easy penetration of the host egg. Although transfer is possible holding the micropipette freehand, a low-power micromanipulator is an advantage. The Singer instrument used here reduces hand movements by a factor of 5. A micropipette is attached with fine polythene tubing to a microsyringe assembly, which is actuated by a micrometer head for fine control. The whole assembly, except for the fine shaft of the micropipette, is filled with mineral oil. Disaggregated cells from an early gastrula are spherical and look much alike except for some variation in size and pigmentation, as this disaggregate from a whole embryo demonstrates. Picking up the donor cell for transfer is a critical part of the technique. If the pipette is too large for the cell selected, as seen here, the cell will pass easily into the pipette and will be injected into the embryo intact. The unbroken cell membrane will effectively isolate the nucleus of the egg and no embryo will form. If the pipette is too small, the membrane will be broken, and the cell cytoplasm that protects the nucleus from the injurious effects of the saline solution will be dispersed, and the nucleus may be inactivated. Using a pipette of the correct size, a cell can be picked up without disturbing the cytoplasm. Yet the membrane of the injected cell will have been broken, so that on transfer the nucleus may be liberated into the egg. The host egg is held steady by forceps grasping the jelly. To assist entry, most of the jelly will have been removed from the animal half of the embryo. It is aimed to place the injected nucleus roughly in the middle of the pigmented half of the egg. Once a pipette has entered the egg, the donor cell is no longer visible, so a marker, a small air bubble, is used to guide the expulsion of the cell. It is visible as a bright spot. The bubble is moved about 2 cell diameters down the pipette. And after injection, the pipette is carefully withdrawn, taking care not to fray the edges of the wound. Healing is rapid, there should be little loss of egg substance, and the cleavage of the egg is treated. A donor embryo is dissected, and a piece of tissue placed in disaggregating fluid. A single cell is picked up in a micropipette of the right size for transfer into the enucleated host embryo. The development of the egg is observed, and any abnormalities are recorded.